Gero, wir hören dich nicht. Gero, hörst du mich? Wir hören hier nichts. Und du müsstest noch den Kandidaten vorstellen und so. Hermann, Dirk, hört ihr mich?
Ja, wir hören dich, aber Gero sagt da gar nichts.
Gero ist plötzlich weg.
Er sagt auch nichts. Er wartet.
Warum ist jetzt
Er hört uns nicht.
wir sehen schon die Präsentation hier, das wäre ja noch eigentlich zu früh. Gero, hörst du was? The problem is the head of the committee obviously doesn't hear anything right now.
I just fixed it. I'm sorry.
Okay. Now you have to start the whole thing.
Yes, I was waiting for everybody to be ready, but obviously my speaker didn't work. Okay, so is everyone ready to start? Yeah? Okay. So welcome everyone to the thesis defense of Ringo Schwabe. This is a very special occasion as it is a joint thesis between the University of Santiago and TU Freiberg. And accordingly, I've been told that we have special visitors today. So we are honored by the presence of Dr. Alexis Aspey, Vice Dean for Research, and Dr. Rodrigo Vidal, representing the PhD program in biotechnology. Before we start, please let me briefly introduce the committee. Because this is a joint team, we have four referees. The Professors Levikan and Ortiz Calderon from Chile, and Schlömann and Tischler from Germany. Dirk Tischler is now at Bochum, was previously at TU Freiberg as well. Further members of the committee are the Professors Nicolai Gonzalez and Gonzalez Canales and Professor Heilmeier from Germany. My name is Gero Fisch. I'm an inorganic chemist, and I have the honor to chair this thesis committee. In the case of this joint PhD, we will follow TU Freiberg regulations for the defense, and I will try to guide you through this as well as I can. I will start by briefly introducing the candidate, and then the candidate will have 30 minutes to present results from his PhD research. Ringo Schwabe was born in Annaberg, which is in the Mountains, not far from Freiberg. He studied applied natural sciences here in Freiberg with a major in biology. And since then, he has worked on biological methods of method extraction in the group of Michael Schlömann, but also with research stays in Chile and with Alemania group. And this will today come to a conclusion with the defense of his thesis. The committee has met defense and has decided that the thesis is appropriate and can be accepted. And now the defense thing that is missing. So then we've reached the point where I hand over to Ringo Schwabe to give his presentation. Please, you have 30 minutes to present your results before we come to discussion.
Hello, I want to salute the audience. Nice to have you here all today together. And the whole general aim of my project is to use metallophores from selected actinobacteria for the extraction of metals other than iron from aqueous phases, and to extend their unique properties for phytomining of strategical elements. So why should I do that, and why it is important for us? Yeah. Maybe. Yeah. Okay, why it is important for us? Clean energy concepts are heavily important or heavily connected to the supply with high-tech and heavy metals, especially rare earth elements. Especially rare earth elements. So it's from increasingly importance also to recover resources from secondary resources and also from waste effluents, industrial waste effluents, and further to remediate contaminated soils and waters. This is from increasingly importance. Also, according to the OECD, the linear economies of humans are expected to pose significant challenges in the next years, as we also use rare earth elements and fertilizers, and the overuse of fertilizer and pesticides significantly interfere with bacterial ecological ecosystems. Also, plants are integrated in that, as metals can accumulate in the food and further through the food web also to the human being. So the problem is that currently recycling and remediation approaches are not sensitive enough for substrates with high volume and low concentrations. So therefore, we use bacteria that have established metal resistance and tolerance mechanisms, which we can use in bioremediation. Bacteria can biotransform metals in form of biodegradation and biosynthesis. They can also solubilize metals from insolubilized minerals or from solid minerals. They are in the integrated in the bioprecipitation, in the bioaccumulation. It's already known that some kinds or members like Bacillus cereus are isolated from environments that are contaminated with rare earth elements, can accumulate these elements. And further, it's well established that bacteria can biosorb heavy metals by different mechanisms. And all of these processes work together in the bioprecipitation and mobilization of soil particles. And one outstanding mechanism, which I want to talk today, is connected with a molecule class called siderophores. Siderophores are metal chelating agents that majorly connect or majorly complex iron. So they are integrated in the iron assimilation, but they also further integrated in nitrogen fixation, as they are integrated with molybdenum and vanadium uptake, which could help key enzymes for the nitrogen fixation. They also mobilize and uptake or help to uptake heavy metals for or immobilize also heavy metals outside the cell. And they are majorly contribute to the biogeochemical metal cyclings by mineral dissolutions and offer therefore a survival competence for the producer. They also can have antimicrobial and biopesticide potentials. And in the last years, they are used also in research, siderophore research, for the establishment of environmental applications. It is well known that siderophores are often produced as multiple siderophore suits, which allow the bacteria to interact on changing environmental conditions. But this is also goes with an analytical challenge, as siderophores are mostly produced in short concentrations, in micromolar until millimolar range, and therefore the isolation and overproduction of these molecules contributes herein in this thesis to make them commercially available. So we need to establish an approach which allows us to overproduce these siderophores, to detect them, and to apply them. Okay. Siderophores are also bind other metals than iron, and there was done a lot of research that they now are clearly established a function that they not only bind iron but also other metals. They previously bind iron 3 to iron 2 in order of in magnitude of order different, and it has a simple biological reason, as it is more difficult to design a ligand which is not selective for it's more simple to design a ligand with iron 3, as there are not such a mass of divalent positive ions such as copper or also zinc and calcium, which could compete to the uptake of these essential elements. And there are also analyzed that metallophores can bind rare earth elements, but it's currently not known or not a lot investigated how these bacteria are interfered with rare earth elements in complex systems like the rhizosphere. Here further I concentrated on actinobacteria, as they are known to alleviate metal stress and growth and support plant growth in different established mechanisms. But the effect of rare earth elements is currently not investigated, especially for members like Gordonia, Arthrobacter, or Rhodococcus. And in my thesis, Gordonia is a model organism which I used to investigate these interaction. Further, I applied that to two other genera. So this thesis aims to contribute the discovery of metallophore mixtures produced by currently unexplored actinobacteria belongs to the genera Arthrobacter, Gordonia, and also Cucuria. So my whole idea is, how can we identify, characterize, and apply metallophore mixtures for metal extraction and phytomining? And thereby, a lot of different hypotheses and research questions I had to integrate to solve this complex research approach. Once you have established approach to overproduce siderophores, I was interested to bring them on a carrier material, which makes them reusable and which could modify and enhance the metal binding molecule or metal binding processes. So it's well known that amino functionalized molecules can bind, or molecules of these foams, of these silica-based foams, mesocellular foams, called, to bind iron and also copper. And my idea, together with Professor Dr. Dirk Tischler, was to immobilize the siderophores with a linker covalently and analyze an adsorption and desorption experiments also other interesting elements like the rare earth elements. So, yes, but I have a lot of, I had a lot of animation inside. So the next idea was to make a phytoremediation with isolated metallophores, as we know that roots can excrete exudates like carbon sources for bacteria. And so the bacteria can be stimulated for their growth to produce siderophores, which can mobilize mineral dissolution and also enhance the nutrient release. And the hypothesis was that we also can use with the siderophore mixtures and enhance root colonization and the majorly point to enhance the transfer of rare earth elements and uptake into the leaves, according to phytomining. So there are a lot of research questions. Are there differences in mineral dissolution from the siderophore supernatants, from the referred strain Gordonia rubri Patinga CW2 in dependency of pH? Do these members from Gordonia, Actobacter, and Cocuria produce siderophores? Can they be bioinformatically predicted and verified by transcriptomic? Can we optimize the cultivation or by altering the cultivation conditions and by the implementation of metal stress, the siderophore production? Can Gordonia produce maybe other siderophores when it is exposed to metal stress? Can we, yeah, it's a list of research questions I have. So the research objectives bring them a bit more onto the point. So I want to identify these metallophores, screen metallophore formation under metal stress, do the functional annotation of relevant gene clusters, optimize the biological production, also characterize their metal binding and mobilization, and experiments for targeted binding and extraction of these metals like I introduced, and at least to evaluate and the applicability for phytoextraction of analyzed metallophores for rare earth elements and germanium. So my research design, methodology, and approach is really simple but complex. So we need to identify, characterize, and evaluate our siderophores. And the approach I have established is combined of a prediction, bioinformatical prediction, which can be used with the genus, also the whole genera or one genome. Then we have established detection methods. I analyzed production and extraction. I established methods for purification and at least the siderophore identification of their structures. Siderophores mostly comprised from catecholates, hydroxamates, or also mixed types and carboxylite types. We here focused on these two functional groups, as they bring a spectra and iron binding, and therefore when we use them in combination, we could observe some different effects between these two functions, molecular functions. So for the characterization, I used two different experiments. So the first is the metal mobilization from soil minerals, where siderophores can dissolute minerals in a progressive process. And this was quantified later in ICP-MS, with ICP-MS. So in the second experiment, I established the immobilization of siderophores and make first tests of metal extraction. And in the last experiment, the evaluation of metallophore coordinated accumulation or assisted accumulation of rare earth elements and germanium by forbs and grasses, where we put in selected bacteria in different substrates and also the siderophores complexed with metals to test if we have on sand or on soil a different uptake of these metals. The procedure I used to analyze the interaction of rare earth elements with metallophores from Gordonia rubra patincta, I will go to explicate you a bit. So the outcome of that, and we started with the genome-based prediction from the siderophores of Gordonia rubra patincta, which is published in these two mentioned papers, and the proposed siderophore biosynthesis of Gordonia rubra patincta includes the four main genes, and we can use this prediction, which is called of an algorithm from anti-smash, to find the siderophore clusters, which are generally integrate all of the functional biosynthesis which is needed. So they start with protein, which is a decarboxylase, and monooxygenase catalyze the next step. So we have then an N-hydroxycadaverine, which is then further functionalized by a transferase to bring in an acetyl rest or an succinyl rest. And the last enzyme does synthesize them and make from the different precursors, which can be linear and also cyclic, different siderophores. These synthetases is NRPS independent. It's not also used with, it's independent from the ribosomal biosynthesis, and also independent from these highly functional enzymes. It's another enzyme, a bit shorter, but do the same as, for example, used in other strains. So we have done RNA library and sent that under iron limitation and with iron, and we can evaluate with this transcriptomic analysis that these clusters are overexpressed and transcripted. So Gordonia produce hydroxamide siderophores, and we now wanted to detect them, and therefore I used the chrome azural S assay. And in this assay, we use chrome azural S, which is a red color, and which builds a blue complex when it is complex to iron. And the iron can then, if you add a siderophore like desferrioxamine B, be in competition with the chrome azural S, and therefore you have a decreased signal. And this is what we can quantify. And we also can use that we can quantify them, and make a calibration curve with them, which is really nice. Also has a nice statistical R value, nice correlation. And we have done this also for solid Cas agar, and there I also normalized the colony area as bacteria on plate have a different areas. So I later make a double threshold for the background and also for the color of the halo that was formed. And after that, you can see that the ANOVA analysis showed us really interesting different responses. So for the Gordonia rubra patincta, for example, we see with germanium and higher siderophore production. And for Rhodococcus or Erythropolis S43 or Rhodococcus opacus 1CP, we also see that lead is inferring, and also gadolinium, like in rare earth, which is enhancing siderophore production for that strains. So I analyzed then in cultivation-dependent production the siderophore synthesis with the Cas assay, and find out that we need to use succinate to have the most siderophore production. I find out the time window when I need to harvest the cells, as siderophores also are biodegradable, and over time the activity is increasing and afterward decreasing. So I used here succinate for the cultivation, and I also tested different metals in the cultivation. And what is really interesting is that when I use zinc or vanadium, and also when I use erbium, then we find growth which is enhanced under iron limitation, and also a bacterial siderophore production. This is enhanced in that process. So that means it looks like that erbium enhanced siderophore production for Gordonia rubripatinta, and with that in mind, we have then extracted our production with XAD extraction. So this was equilibrated with the resin, and afterward we diluted with methanol to obtain an crude extract, which was further evaporated to a shorter volume. And afterward we use a separation with high performance liquid chromatography, and therefore we have established different, or also a calibration curve with desferrioxamine B. So we back for so here you can see the program or the profile running. Can you switch one here? That's nice, thank you. And so we have a calibration curve which serves, and we have established the program, the HPLC program. And so we have a separation of 3 peaks. When we look for DFOB and Gordonia rubripatinta, it seems that Gordonia rubripatinta really produces hydroxamide siderophores like desferrioxamine B, DFOB. And also we can separate between metal-loaded siderophores and siderophores which do not have binded more metal. And so we analyzed all of these fractions, and further I have done an evaluation where I can see that when I use this procedure with HPLC fractionation with an C18 material, we have the highest concentration of desferrioxamine B. When I use liquid chromatography or an FPLC, we only extract slow concentrations of them. And after storing the XAD extract, you also lose siderophores by degradation. And also after 1 year of storage, lyophilized siderophore extract, we still have some siderophores there. So it's not that it is everything degraded. So, and then we used the metabolomic approach, which was established by Christoph Senges, Dr. Christoph Senges, for the structure identification. And therefore, we make the production, the XAD extraction, the HPLC purification, and send them then to Christoph. And Christoph detected after the separation of the metabolites in the mass spectrometer and the fragmentation of them, different patterns which were compared with databases to find similarities in these fragment pattern to build up molecular networks, and from them we can extract the siderophores, which has built mostly by Gordonia, in that case citric acid, which is also called carboxylate siderophore, but it is one of these smallest ones. This ferrioxamine E, a cyclic siderophore, and his short product is succobarin B, and also a ladder of different desferrioxamine B equivalents. About 28 different unidentified metallophores are still to investigate, open for another research. And I further investigated the metal selectivity of bioabsorption of adaptations. When I grow Gordonia under mangan chloride and also under erbium chloride conditions, then I can see that when I transfer the plates to gas media, I saw that one strain or one colonies were produced a lot of siderophores, and these ones I have dried frozen, and with these dried frozen preparate, I have added a multi-standard with different elements, and then used with ACPMS and before an acid exploration to compare how or can we see a difference after adaptation. And yes, we can see a difference after adaptation. Really interesting is that the wild type binds better the iron than the adapted one, which binds better rare earth elements. So it seems that we here in the CAS assay see a positive siderophore producing microorganism, but it can also be the case that it is only an enhanced succinate or citrate production of the strain, like an adaptation process, which is referred to metal tolerance. And I induced an approach where I used siderophores for metal stress and used or measured the pH profiles. And we can see later when we compare Gordonia, the yellow one, with the manganese, the orange one, then you see that is a higher or a faster response on pH, and all of these are increased until pH 9. I further used the different established manganese adaptation to analyze also these molecular networks. And we see that when I add erbium to the media in the cultivation, they will be produced or were produced equal or similar patterns of the metabolites, but which are definitely distinct to the molecules which we already know. So we do not have investigate them currently until the detail, but we see these differences also in the number of unique MSETs and also in the retention time. So they released a bit later in retention times when we used the wild type strain, and also in a bit higher intensity and MZ values, which is like a post-translational modification of these siderophore patterns. And the same we can also see for the adapted for the adaptation. They already have these high retentions, and when I use erbium of that, we see that there are fragments there or patterns there that are extremely induced with this procedure of adding erbium. So to see also in a more realistic way how siderophores can mobilize these metals, I used batch experiments in different pHs, and here we can see the siderophore function that we have in a higher pH, a higher function of the siderophores. So this is environmental correct, as also iron-free is only existing in low pH, and at neutral pH, it's really low concentration. So siderophores are needed to enhance at these neutral pHs the solubility of iron, which we can already also see here on the data. And the manganese is in contrast the other pattern. So with higher pH, we do not have a significant improvement with siderophores, but at lower pH, when siderophores lose the affinity for iron 3 over iron 2, siderophores also can complex significantly in comparison to the control, which was used here as a medium control, also manganese. These patterns are really interesting, as for copper they show the same, and for zinc, and also for gallium and germanium. Always these patterns. That means we could obtain, and at lower pH, also selectivity beneath iron for other metal ions. And the immobilization of siderophores, especially the covalent immobilization, showed us that the carrier already binds all of the iron. So the material was not the best choice for that. And we also used these extracts, which we obtained from a dissolution experiment, adsorption and dissolution experiment, and tested them on ion concentration with the same colorful assays. So that we only need to expect here the concentration of neodyne. And we see that the carrier does not bind that hard the neodyme, like more reversible. So it was the first indication that the siderophore of CWB2 could also bind, in my experiments, neodyme beneath other metals. So I have changed the immobilization method to entrapped metallophore beads, which are synthesized in a polymerization in the acid catalyzed hydrolysis and the acid catalyzed condensation. And these I called precursors are then formed networks together, and at the right point you bring in the siderophore with a complex metal. So our idea was to imprint this structure into the gel to get a highly connected network, which beats clearly interconnections. So we have also obtained these clear beads, and which was really interesting is that when we show for vanadium, we have the highest absorption with our concentration of CWB2. When we look at gallium, DFOB is also has the highest gallium extraction, and this is also in literature according to Yain 2019, which has patented an immobilized desferrioxamine on the same material and show that this is able to bind gallium. And here further we see a selectivity for molybdane, and all of the hydroxamide siderophores seems to bind neodyme as a rare earth element. So that was a better experiment or better material for that, where we could go on to establish these different materials and immobilizations, and to test then the evaluation. We set up a pod experiment, like I mentioned before, and for the prediction, here we have used Arthrobacter and Cocuria, different strains isolated by Dr. Gerardo Retamal and Professor Gloria Levikan, and they are coming originally from a mine in Aue in Germany, and I used these strains to optimize or to transfer the knowledge to phytomining. And I also use these genera related functional annotations and search the whole database of anti-smash for blend interacting molecule classes, and the high diverse classes, the siderophore class for these 4 strains or 4 genera, and all of them can produce hydroxamide siderophores, and especially heterobactenes for Rhodococcus and desferrioxamines for Arthrobacter, Cocuria, and Chryseobacterium. With that analysis, further we analyzed with the solid CAS assay their efficiency of ion complexation, and I selected Arthrobacter oxidants ATV2 and Cocuria ATV4 at the best ion chelating producers or siderophore producing bacteria, and I used then these in a next soil mineralization dissolution experiment in the same manner like I told you before, and Arthrobacter ATV2 and so for phosphate and gallium, for example, but also for silicium and germanium, better binding or mobilization capacities, and Arthrobacter ATV4, especially for iron and for rare earth elements. Therefore, I have analyzed, I have selected that. Here we see the ANOVA analysis. All of these methods are already published in Chemosphere. And we make the structural analysis of them. So
I'm sorry
we
to
find
interrupt.
out that
You
they
already
produce
have a bit over your time. I have to ask you to finish soon.
Okay, can you give me some 5 minutes and I will conclude everything.
Okay.
Thank you very much. So, and you have different patterns when you use a tissues accumulation analysis, and these patterns are definitely really interesting, as they here are presented with nutrients and these rare earth elements, but we can't go for all of that now, so I will summarize the effects of that. I have done this pattern analysis and tried to figure out the different effects which have the inoculation. And you have to do that for the 3 different plants we have tested there. And what we can see, Brassica napus also, beneath Phalaris arundinacea, Iavena sativa. And the effects you can really see is that we can enhance the germanium accumulation. Significantly for the rare earth element accumulation, significantly for the strains used, and also for the metallophore procedure or thread I used, and to record Gordonia rubripatinta produces siderophores. We have analyzed and verified the genome or gene cluster with transcriptomic. We have optimized the production with succinic acid. We see that they interfere with neodymium, erbium, and zinc. We have seen that the pH profiles of them are different when I use the adapted variant. We also have seen the PhD dependent biomobilization of them. And I showed you the differences of the secretome and biosorption. Which all could lead to, and also the metal extraction of metallophores, which could lead to another process of a sustainable metal extraction. But we also see that the carrier binds a lot of iron, so we should change the carrier to material which does not interact with such that high adsorption. And we could also think about other processes which we can use, like an electrowinning, where we can desorb the metals directly in form of a reduction at the surface of the bead. And we saw in the metallophore-assisted phytoextraction that plant-specific element uptake and transfer, which was indicated from our treatments, the bacteria have specific plant growth promoting effects. The metallophores also have specific effects from the surfactant. And then we also see a substrate-specific adsorption of the elements. And we have the metallophore treatments affect the root accumulation of the plants. And also metallophore and surfactants can enhance the uptake and transfer of rare earth elements and germanium, which is especially plant-specific. So all my papers are published, and we are only at the beginning to set up an approach which is combined on metallophores and on biorefinement, or phytomining and biorefinement. And what we need to set up more in the future is to expand the experimental design on the whole rhizosphere, analyze also the plant exudation, detect the rare earth interacting bacteria by metagenomic, isolate the rhizosphere key player, do the substrate-specific mineral dissolution experiments for this key player, combine also the endosphere and rhizosphere key player together for treatments, to have a microbe and also analyze the microbe-plant interactions, which can further be predicted by genome and transcriptomic approaches. So I want to thank all the people which has helped me to realize this project, especially Michael Schlömann and Dr. Dirk Pischler, where I have done the first 3 years in Germany, all the experiments, and further Professor Heilmeier and Dr. Oliver Wische, which integrate me in the next project for the phytomining and Phalaris 2, and which allowed me to go to Chile to make here in the laboratory also for Professor Gloria Levikan all the rest stuff which was needed to analyze these new strains. Thank you very much. So I will salutate the audience, and I'm excited about the questions which will follow.
All right, thank you very much for your presentation. We get then to the discussion. We are quite a big committee, so I will have to ask everybody to keep their questions relatively short. The tradition in FIBAC is that we go by the following order. We start with, in this case, the 4 people who have written a reference and evaluate the thesis. Then we will proceed with the other committee members, and after that, the talk is open for discussion to everybody else as well. I will keep minutes here of questions that have been asked. And for the professors who have written a reference, I would also ask them to say like one or two sentences about the thesis, if they wish to. Right, then we start with the first supervisor, please.
Now on? Yeah. Okay, maybe I do it from here so that people see me. As we have heard, Ringo has done a lot of work and work in different areas, from genome analysis, isolation, characterization of the metal extraction, isolation of the compounds, structure elucidation, attaching them to immobilize, use to grow plants and investigate the uptake into plants. There is a number of different results and corresponding publications, so quite some outcome. Yeah, that would be my my statement, and maybe I can ask a few questions. For that, I turn to you. One question would be referring to the purification of and the structure analysis of the siderophores. You mentioned that there are suites of siderophores, so not just one, but several. And I wonder whether the approach you have taken is really optimal. Wouldn't it make sense to do the LCMS directly after the solid phase extraction?
Thank you very much for your interesting presentation. And yes, you are already right. The approach has to be a bit optimized, as when we extract after HPLC fractionation and analyze all these extracts, we will only get these analyzed peak, which we can extract, but we do not have before separation and purification all of the metabolites we will lost. So a more general view on the siderophore extracts will be done when we only use XAD extraction. But this comes along with analytical changes for the LCMS. And at least for other structural identifications like NMR. You need to have a purified metallophore, which also do not bind iron. But for LCMS, it's maybe sufficient to have the XAD extraction and to analyze the whole siderophore suite, which I mentioned. This is what I have done, not for the Gordonia only, but also then only for the other strains tested. So therefore, we see that we have a combination of bacillibactin and also desferrioxamine siderophores, which have catechol functional groups and hydroxamide functional groups. And also we see that this one strain of them produces a biosurfactant. And so to come back to your question, when we use the approach only with the XAD, we have a picture which is more rounded. So yes, it should be done at that point. But to have it clear and purified, you also need the extraction method with HPLC.
Okay. You mentioned that there are suites and that the bacteria could have a chance to adapt to certain conditions. Are there examples that this is really being done, like that a different pattern of siderophores is made with different metals or different iron concentrations or something like that?
the production of metallophore suits is oftenly or obviously dependent on the enzymes that make the biosynthesis of these molecules. And these enzymes have called enzyme promiscuity. That means they can accept also other enzymes which are not the best, other substrates which are not the best for the enzymes, like precursors which can differentiate. So currently, there are not many examples how these enzyme promiscuity are used in nature to react in relative short times to changing environment. So this would be a next research challenge to monitor this process of metal tolerance, which have these bacteria with production of different enzymes, different siderophore mixtures.
Okay, and my final question concerns the adaptation. You showed results that there is an adaptation to manganese and erbium. What is behind this adaptation? My question aims at, is it a mutation or is it induction of certain enzymes? And if it is not clear, how would you find out?
According to the adaptation, I have sent a genomic DNA sample to also analyze the genome of this adaptation. And we find out that there was no mutation, but we was not sure to find one point mutation in the genome is not the simplest way to do. So I'm not sure if this was a mutation because the erbium chloride, which I introduced on the plates, is known in literature that they can introduce mutations, which are based on the polymerase interferation. So they can interrupt polymerases, they can make some errors, and then you obtain with this metal stress some mutations. So I was aware to not say that's a mutation. So I talked about adaptation, as it was not clear. But a mutation can be introduced by mangan chloride. Thank you very much for your interesting questions.
All right, thank you very much. Thank you very much. Maybe can you pass on the microphone to Gloria Levikamp, please?
I hope. Okay, Ringo, thank you very much for your presentation. It's obvious that there are huge amount of results, sometimes difficult to follow, but it's obvious that it's a big work. I have just two short questions. And the first one is related with some statements that you mentioned about some antimicrobial property of siderophores. So how does it can happen? So what is the activity in this case, or which is the phenomena that is involved in this?
In the siderophore production?
Yeah, no, you mentioned that siderophores are involved or have some antimicrobial activity. So that is not evident how that.
Thank you very much for the interesting question. So they can also have antimicrobial functions with a mechanism which is already known called the Trojan horse effect. That means they can also bind metals which could be toxic for the pathogen, and the siderophore could introduce as a complex, metal complex, these metals to interrupt the grow activity of pathogens. So antimicrobial action can also happen when better siderophores produced by a neighbor of a pathogen, and that can uptake the iron more efficiently than the pathogen. So the survival competence of of good bacteria is increasing of that.
Okay, okay. It's like sequestering the metal for the pathogens.
See,
For example. Okay.
exactly. That's
Okay.
it.
So the next question is something related with Professor Michael asked. Because in case of iron, siderophores are usually produced when there is iron starvation, not? That is what is described. But in the case of other metals, some examples, the siderophore are produced when metal are present. So not always is the same pattern regarding to the regulation of the synthesis. So what do you think about the role of siderophores regarding to the other metals? Are related with nutrition or maybe with other phenomena?
Thank you very much for that interesting aspect. And the siderophores could be for bioassimilation of metals, but also for immobilization outside of the cells, means a protection system. As the highly recognizable or the highly efficient recognition at the surface is controlled in the first instance on membrane proteins, which needs to find the complex with metal and siderophore from their redox potential. You have siderophores with different redox potentials, and when they complex different metals, you have a different signal for these enzymes. So it could also be a mechanism to produce siderophores that could hold these metal complexes outside of the cell. So you should see the metal from different perspectives.
Okay, okay, I understand. Thank you very much.
Thank you.
All right, thank you. All right, thank you. Maybe we can continue with Claudia Ortiz Calderon.
No. Thank you, Ringo, for your presentation. It's a lot of work, as everybody has said. So I think just to mention that you have to be a bit careful with spelling of some words in your presentation and also in your manuscript and then your thesis. So have a look at that. Well, I have a question related with the phytomining. Phytomining is a very interesting field, but also has got a lot of like limitations. So in that sense, can you discuss about in your model? Yeah, let's talk about the rare earth metals. What do you think are the main limitations and advantages related with or compared with the traditional methods of rare earth metal extractions of this model that you propose for phytomining?
Yes, thank you very much for your interesting aspect on the presentation and the thesis and phytomining especially. So phytomining is, as you called, has a lot of different limitations. It's about plant-specific uptake, it's about the substrate, it's about the rhizosphere ecology around the roots, and we has to drive through different impacting factors. So our idea in the system was to use a system which is really limited. We do not have used hypoaccumulator plants, which can taken up different metals, at majorly nickel, as we wanted to see how do energy crops react on that. And because if you use an hypoaccumulator, the plant growth in general will not that much the yield after a period of cultivating the plants as you use normal plants. So this would be improve phytomining when we find or when we can also use for non-hypoaccumulator these cytorophore and cell treatments to optimize their metal uptake or their nutrient uptake, and to make them grow also in contaminated lands. So we have we have in plant limitation on the uptake. So the plant has also different mechanisms to, you have strategy 1 plants and strategy 2 plants, which also then can actively import iron and other nutrients. So this is one point of limitation. And we wanted to make a synergetic effect when we use cytophore producing bacteria that are also plant growth promoting bacteria. And as Arthrobacter already was known, and also Cucuria, to help the plant to grow in contaminated sands and soils, we used exactly these ones as model bacteria, as we already know there are mechanisms behind that. So
okay, thank you. Yeah, well, you have to consider the whole ecological system. I mean, it has to do with the relationship with the bacteria, also with the plant and the plant.
Yeah.
It's very specific sometimes because you can have a negative interaction, which is not good for the plant, for instance. So that's what I wanted to address.
So we are worked with that model system at a really limited way in the corner to see can we bring some effect of cytorophores inside of that.
Yes, and a second, it's not a question really. I would like you to highlight briefly and just like in a kind of synthesis. If you have to highlight the main findings of your work, which ones are? What is the main highlights or findings that you can say, this is what I can contribute, or with this I contribute to the knowledge in the field?
Yes, thank you very much. So the highlight of my thesis is, as I told you, Gordonia produces only citrate. So in the first beginning trials, I have analyzed and I have, for the identification, the production of this strain, Gordonia especialmente, or special. So I analyzed all of these metal stress ingredients and put out that rare earth elements can induce cytorophore production. So one highlight is that we can obtain cytorophores when we have in metal stress, which is a new revenue to obtain different biological molecules, which are also for medical interest. This is one highlight, the approach to identify cytorophores also from producers that at first only produce citrate. So I have optimized that and get out that they produce also the expected cytorophore suit, which we has theoretically proposed. So the next aspect is that I have overproduced them and immobilized them to a material or two different materials until now to show if they really have this function when they are stabilized. And yes, they have that. So we have extracted some neodymium as a rare earth element, which was the main point at my title of the thesis. And this is also one point of importance. And the third important point is that we can also increase in plants the metal uptake when we have also plants that are not hypoaccumulators, when we use bacterial or bio-augmented phytoextraction. And that in the background of their metabolomic structures, of their functions, and also of the genetical potential of these strains. All of that methods together make us able to have a new perspective on metal binding characteristics of the strain. So before my thesis, there was only these colorful CAS assays, and I can add to the scientific community also this immobilization process, which allows us to characterize metal binding, to select promising bacteria better, because this is one of the key factors in establishing biotechnology technological methods, the selection of promising bacteria.
Thank you.
Okay, thank you for the questions and discussion. We continue with Tischler, please.
Yes, thank you, Ringo, for the presentation. I'm happy to see that this long journey and so many aspects of cytophores is finalized in such a nice presentation. So I have some questions for you. So Gordonia, as you said, produces mostly citrate and some desferrioxamine. If we focus on the desferrioxamine, the gene cluster is rather small, but it produces about 15 to 20 secondary metabolites from such a small cluster. Can you explain or why a cluster of only 5 or 6 produces so many different compounds?
Hello, Dirk. Thank you very much for your interesting presentation. And yes, it may be wondering why you can produce with such a small gene cluster such a mass of different enzyme siderophores. But as I mentioned in one of the first questions, I think from Professor Schlömann, siderophore biosynthesis is mostly done by enzymes. And these enzymes do not have a substrate specificity which is extincted or distinct. So they also allow different substrates. And the concentration levels inside the cell of potential precursors is mostly affecting the enzyme mechanism of biosynthesis. So if you already have stress outside the cell or something, and you have a response of the cell, then the cell could support the mechanisms that they can use already existing precursors to make different siderophores than the expected ones. And so the cell have an adaptation mechanism based on the enzyme promiscuity, which allows to produce different siderophores in dependence of the level of precursors which are already inside the cell. But as I pointed out with Professor Michael Schlömann, we already do not understand this point of relation, because we only can measure siderophores outside the cell, but not inside. And this is another research challenge which could be taken.
So if you could manipulate by feeding, what would be a substrate you like to feed?
For me, it would be nice if it has a color, because when it has a color of the substrate, we can analyze and detect them rapidly. So, but in the front of or in the background of the environmentational significance of that, we should use precursors, or we should use the contaminants which already exist in the soil, which we want to remediate. So this would be, for me, more interesting than use a colorful aryl siderophore, which we can detect rapidly. But it's also more challenging than that.
So
yeah, but a colorful thing would be nice.
Maybe a different question. So if you compare the citrate and the desferrioxamine, so basically we know all about how to produce citrate, and citrate is also a siderophore. So why do we not just improve citrate production? What is the beneficial thing about desferrioxamine?
The beneficial thing of that is also the point of view, where you, or what you want to do with the siderophore and citrate mixture.
Okay,
in
then
terms of bacteria.
Before you answer, okay?
Yeah.
So you have chosen bacteria and plants, and if you think about mobilizing nutrients or metals for phytomining, I think citrate is a very interesting compound, and I don't see really a beneficial of desferoxamine. Why would you still improve desferoxamine production?
I think when you have a mixture of desferrioxamine, which has a really more huge affinity for iron, and a citrate, which also do complex different metals unselectivity. You can, or you should keep in mind, you want to have something selectively complex. So more would be complex from the desferrioxamine B, or for a longer period, as for the citrate and the equilibrium constants regarding to the metals. And so when you also expand your idea to different structures of siderophores, then you can adjust maybe according to the environmental conditions such as pH, ionic strength, and the contribution or of metals, the abundance of different metals. Then you will be able to change a pH and to change the metal binding, the selectivity of the metal binding. So sure, you can use citrate to mobilize everything, but when you want to take up the citrate, or maybe if you want to not take up some metals, then you can add in siderophore, which holds these metals outside of the plant root cell. And also we see that in my experiment on phytomining, that when we use a siderophore mixture of catechol siderophores and hydroxamide siderophores, especially when we also have surfactant inside, then you have a higher root accumulation of rare earth elements. And when you then have also complex system like citrate, which can bring in the rare earth elements, then you can enhance the accumulation of them. So you can accumulate at the root outside the cells, especially rare earth elements, and then you can bring in them. So you have synergetical effects when you use at the site of citrate different siderophores. And not only one, a mixture would be great. Yeah, that's my point.
Thank you,
Thank you very
Ringo.
much for this interesting.
Summarize diversity is good for nature. Thank you.
Sorry, I do not have here listened. Which was your question?
I'm done.
Can you repeat, please?
I think that was the last question.
Oh, okay.
All right. So this concludes the questions of the referees. We move on to the questions of the other committee members. Maybe we can go with Professor Godoy Gonzalez next.
Hello, Ringo. Thank you for your presentation, for your interesting result. I have some comment about the inorganic chemistry point of view. I want to talk some comment about the chelating capability, selectivity. You say why was just the pH dependent mobilization in the range from the 5 to 8 studied and no other value? You study only maybe 4 pH, but in the case of the SAM diagram of the iron tree, you have different species, hexago, and one ligand hydroxyl, 2 ligand. And when you analyze the result. In some case, for example, lead, you have increasing of the amount of the lead with the second bacteria, and then one of them is like a plateau, and the other is increasing maybe 4 times. What do you think about the species, about the result obtained?
Thank you very much for this nice, interesting aspect on the work and for this dissolution experiment of minerals. Yes, we used physiological interesting pHs where the bacteria can live, where we can show these effects in that pH manner. So it was only to choose more than nothing and physiological aspect.
Maybe it's difficult the question because it's have a concept in organic chemistry analytica, but to talk about the Pearson theory, you have acid, strong and hard. If you had chart 3 for the hexaco complexes, and then you put or replace one water for one ligand hydroxide, the chart is reduced, and it's different the capability of the coordinate. I don't know if you know something about your molecule is a long ligand, have a many functional groups, is fluxional and can adopt octahedral structure.
And the interesting point is for that experiment, we do not have only used the desferrioxamine B, which is a reference siderophore. We also used an natural extracted solution of mixtures of siderophores. So of course, it's harder to follow which species of ligands we have found at the pH as we have mixtures. And another siderophore, for example, bacillibactin, which is a catechol siderophore, has a more rigid structure. It's not that flexible
exactly
as it can do by desferrioxamine. And that means also, referring to the literature, Bellotti et al. 2021 show an homage for desferrioxamine B that can bind 20 different metal complexes or in a stable form with stability constants higher than for citrate and magnitudes higher.
Yes.
So you know that would becomes complex, the thing, when you add a mixture of different siderophores. We also used a producer a bacteria called Paracoccus denitrificans in that experiment. And this was another, he produces mainly putrebactin or peptide siderophores.
Yes.
And therefore you have another complete and another equilibration of the metal binding of that. So it's hard to say why it's at that point higher with the other strain, as we do not have done the metabolomic analysis such detailed as for the other strains. But we should keep in mind, addressing your question, that siderophores are flexible, and there are also structures which are furroundingly binds iron. And lost in pH decrease, they are function of iron binding. And bacillibactin, which has really high iron affinity, becomes equal like citrate for chelating iron when you decrease the pH. So these are these effects we have to solve with these kinds of experiments, but therefore we need to add more theoretical knowledge, modeling, and also I added the Popo diagrams will be useful, but not will solutionate the problem or the question of it.
This is important because when you change the pH value, you can change the species, maybe a positive, neutral, and negative. And this is opposite with the chart of your ligands. This is, I think this is important consider with the Purve diagram. And the last question is about the UV visible spectrum. You have one of them is zinc and the other is maybe lead. I don't know if you remember. You have two spectra. One have zinc and the other have lead. I don't know if you remember. No?
I have measured a lot of spectra.
Let me.
And which paper would be?
Iron and zinc. You made the spectra in the for separate one spectrum for
Okay,
zinc.
for the purification of
Yes.
publicated. Yeah, okay, I got.
But you had an important band, metal ligand Charles transference, NDD. When you have the complex, the iron, you have one band around the 430 Celsius.
Yeah.
What happens if you put in this solution maybe a zinc, maybe 100 or 1000 amount if you have selectivity for zinc or for iron?
Thank you very much for the interesting presentation. I have done such an experiment where we measure the response in form of fluorescence quenching. And this was what we have also obtained, that always the siderophores have some amount of iron bounded. It's hard to differentiate them. So when we add a metal like iron, we get in fluorescence quenching and decreasing.
Yes.
And when you add in manganese or zinc, you get an increasing.
Yes.
But you can't hardly say without more investigation, like titration or something, to see which ligand ratio we have now, according to your ultimate question. So I can't say at which ligand we have this effect, where we have an increase or where we have a decrease. But with fluorescence quenching, this is what you will see.
The physicochemical response when you have maybe 10, and then you put some amount of the other interference, 110, 100,000, this signal must not change, maybe 5%. If change is interference, you can say something about them, but for the future. Thank you.
Okay, thank you very much.
Okay, thank you. Then we move on to Professor Gonzalez Canales, please.
Hi, Aisem. Following the same context from the question from the Professor Dirk about the number of the genomic detection of pathway associated with biosynthesis of these metal offers, and the number of structures that you could found in their analysis, in your analysis. My question is associated with the biodiversity of these compounds. If in this area, you may use a genomic approach. A lot of genomic data are available to understand this aspect, for example. And if you look the genomic data, you may estimate the biodiversity of these compounds. So, but when you analyze the structure, you have other numbers or magnitude of this phenomenon. And the other aspect is that you use in your thesis is the activity, because you also analyze the activity of these compounds in different contexts. So my question is more on comments from you about how do you think about the biodiversity of metal offers in the nature when you look on bacteria? Is the best option is to see the genome, the best option is see the activity in vivo, or look the structure by one biochemical approach?
Thank you very much for this interesting aspect on my thesis. And yes, the best one would be to combine these three approaches. I mean, genomic, transcriptomic, metabolomic. Only in that way you are sure that you have some discrepancy on the prediction and what you get out of your methods. And you always have loss of some kinds. And also the purification of siderophores can lead to a loss of some siderophores. And also when you analyze the structure of them, you make like a puzzle, like a fragmentation pattern from siderophores, which were fragmented in the spectromotor. And so you maybe can also lost some of different siderophores. But what we have measured here is that we get out with a higher diversity, which we has exposed. And that means we have especially for Gordonia rubripatinta, which was exposed to produce desferrioxamines, that they can have also modified desferrioxamines, which are already not known which are of them. So the production and isolation brings in some errors, and also the method which you use for structure isolation. And you also have in laboratory not the natural condition of the bacteria. So you have in laboratory setup, and you choose a distinct carbon source, and all of that is not related to the genome and not related to the information of that in terms of biosynthesis. So the only thing where we can go is to have the exactly enzyme substration specificity or enzyme substrate specificity. When we know that maybe only a kind or only leucine, which is the precursor of desferrioxamine B, for example, or when we know that they can also integrate ornobactine, ornithine, which is the precursor for aerobactine siderophores, then we know better that we have that we can produce or which siderophore suits are possible. Only from the prediction, it's not possible to say which siderophores are produced. So it could be that these 28 new siderophores are also some fragments or some only siderophores which are modified post-translational. So this is,
no, thank you.
Okay, thank you very much. Thank you very much. Last but not least, Stefan Heimer, please.
Oh, thank you. Oh, thank you. I have some maybe very plain questions. So first is when I read your material sections, I was asking myself, what is the kinetics of the production of siderophores by bacteria? Is there anything known on that?
the kinetics of the production of siderophores. Hello, Professor Schlömann. Thank you very much for your interesting aspect on my thesis. And the kinetics, maybe you refer to the production in time. And so when you have a highly genetically regulated biosynthesis, then it's always really dependent on the level of metals which are bioavailable for the cell. So when you make a time profile with a Cas activity test, then you will see an optimum from that, which reach a plateau and then decrease in production. So you have a time window where to obtain the majority of siderophore production correlated with the growth of the bacteria. As the bacteria goes from the exponential phase to the lag phase and to the point of that, then you will obtain them in the moment of limitation as a strategy to reobtain growth conditions. So the kinetic would be highly correlated to the concentration of nutrients which are bioavailable in that moment.
Thank you. One other issue which you mentioned was that in the media when you had experiments on plants, the media contained amino acids and low molecular weight organic acids. And how does this affect production of siderophores?
Especially for Gordunia rubripatinta, which is mostly dependent also on citrate production, anaplerotic sequences or substrates which can enhance the production of some secondary metabolites are all related to the citric cycle. So we can enhance siderophore production at one point when we bring in substrate which already exists and can use for the next step of biosynthesis. So therefore, I also see that when I use citrate or succinate, exactly that the siderophore production is enhanced.
This is true for siderophores which are now citrate. If you add
citric acid,
citrate. Another issue you mentioned that the production is very low. Is set in the micromolar or millimolar range. So I think you know on your own that micromolar is a concentration, not a unit of amount, which means a unit of production. So in which compartment are these concentrations measured? Or do you mean micromoles, millimoles of whatever compound?
I mean that we can, outside of the cell, measure the concentrations of the siderophores, not inside the cell.
But this, of course, depends on the medium, how the siderophores are diluted.
Yes, this will hardly dependent on the nutrients available in the media.
And if you compare the concentrations of the nutrients in the soil solution, the concentration of these concentrations of siderophores, how do they match?
And the bacteria also have a capacity of siderophore production. And as siderophore production is hardly genetically regulated, and also in the terms of how can siderophores with metals be uptaked from the cell surface, you will obtain more siderophore production when there are enough of iron. So it costs the bacteria a lot of ATP to produce special complex siderophores. So bacteria are high efficient to optimize their energy costs, and so they also use these strategies to mix productions. Means citrate production, a lot of citrate, and a bit of siderophores will be the same as you make another case of that. So,
but if I imagine a very small bacterium in the big volume of soil solution, isn't a millimolar concentration of citrate a huge waste of resources? Because it's a bacterium that produces a lot of citrate to dilute it in this amount of soil solution.
Not at all. Clear, if you have a bacteria which only produce a lot of citrate, does not will economically survive. It needs to have an interaction with other organisms that can use these outputs. And so, especially in soil, where plants and bacteria live together at the surface of the root, you can enhance with that sustainable cycle of metal cycle and also of carbon cycles, which means they produce at the end an equilibrium which supports all of the organisms inside. And so a producer, a citrate producer, will maybe found in nature at rhizospheres also, to combine the interaction with plant to realize or to maintain growth also under metal stress. So it would be effective.
Thank you. Thank you.
Thank you very much.
Okay, let me thank all the committee members for their contributions to the discussion, and also for Ringo Schwabe for his answers. Now the discussion is open to everybody. Is there anyone else from the audience who would like to ask a question? This does not seem to be the case. Okay, if there are no other questions, then let me thank everyone again for the contributions, especially Ringo Schwabe for his presentation and for answering all our questions. We will now retreat and consult about the final of the dissertation and the defense, and we'll reconvene here whenever we are finished, usually between 15 and 20 minutes. Right, thank you.
Thank you very much. Ihr selber? Oh, ich gehe. Ich auch was.
All right, all right. Is everyone back? Oh, the echo is back as well. I don't see anyone. Unfortunately. I need some guidance. And I'm afraid I can't hear anyone either.
You can go ahead.
Okay, very good. So Mr. Schwabe is back. Maybe he can come to the talk so you can see him.
Yes, I am back too.
Okay,
We
very
do
good.
not have a. You do not see any of us now.
I
Can
see
we
your
switch
head.
on
Your head.
My head?
Behind.
No. Can
I can see the
we
screen.
turn the camera?
So if someone's standing in front of the screen, I can see them.
We still try to solve the problem.
It's okay. It's okay. If someone is just standing in front of the screen, then it's clear. Okay. So the committee has met, and so first I can congratulate you, Mr. Schwabe, and award you the title of Doctor Rerum Naturalium, so the PhD in Natural Sciences. Congratulations.
Thank you very much.
The committee has looked obviously at your dissertation and your talk discussion. We found that you are very hardworking and also a very knowledgeable student. The committee criticized at some points that you find it difficult to focus and work out the key points of your work, and maybe leave aside the less important parts. That's also an important part of research, that you're able to work out the key points of your work. I think that was also reflected in the events today. You noticed yourself, you overran quite a lot by your presentation. But on the other hand, in the following discussions, when you had clear and concise questions, you gave very good and knowledgeable answers, and we were actually quite impressed by that part. Okay, so bottom line, the committee awarded a grade of 1.7 and hence cum laude. Congratulations again, and all the best for your future science career.
Thank you very much.
Right. So in this case, this concludes the session. I'm sorry I can't just jump over for drinks, but I'm sure you will have a great day. So let me thank everyone again for being part of this. Such collaborations between TU Freiberg and the University of Santiago are very important to us, and they promote the collaborations in science very well. So it was a pleasure of chairing this committee and spending these last couple of hours with you. And I wish you all the best for the future, and we will hopefully have a great party tonight. Thank you and goodbye.
So in our university, we have the tradition that the people get a doctor's hat, which is made by the group of the candidate, not by the professor. And in this case, it was a little bit difficult to transport. So I got a kit prepared by people in the group. And
Nice.
together with Gloria, I put the kit together. So
Thank
I
you
can
very
now
much.
give you this doctor's hat. Oh. I hope it fits. Yeah.
Thank you very much.
Actually, I
Vielen
think
Dank.
I'm not so used to that.
It's
Maybe
also
this
my
way is
first
better.
one.
I want to put this one now from Usas.
Qué bacán, qué maravilloso.
Okay, congratulations.
Thank you very much, Chloe.
Okay.
And then the people usually also get a little book.
Nice, thank you very much.
Congratulations.
Done.
Okay.
Maybe in front of those. Yeah, makes sense.
Nice.
Yeah. Yeah.
Okay.
Yeah. So
You need microphone. Use the microphone. Then be able to listen.
okay. Oh, sorry. I cannot read whether it's off. So you have seen that we can congratulate it, and we are basically ready to leave. Maybe you want to say anything, but otherwise we can switch off.
Glückwunsch, Ringo. Tschüss. Tschüss.
Danke, Dirk. Vielen lieben Dank. Auch danke, Hermann, vielen lieben Dank und auch an Gero, dass alles so super organisiert war. Ja, ich möchte mich an alle nochmal herzlich bedanken, die daran beteiligt waren. Es war ein langes Projekt und jetzt ist wieder genug Luft für neue Dinge. Und ja, schön mal wieder was beendet zu haben, auf alle Fälle.
Alles Gute, alles Gute.
Dankeschön. Auch für euch.
Tschüss.
Glück auf, Ringo.
Tschüss.
Glück auf, Ringo.
Bye.